All right, good morning, everybody. Sorry that took me a second to get this stuff set up. I clicked on Zoom and Intro Micro wasn't there. It was just like start a meeting, and I can't do that. I need to click on the right Zoom link, or all of you guys tuning in at home would not see anything. Okay, but I think we're in business now. So we left off last time talking about the potential for sometimes virus to get into a cell, but to manipulate it in a different way. With transduction, it's going to basically transplant a gene from one bacterium or a chunk of DNA. It could be multiple genes, but it's going to transplant some information from one bacterium into another, and potentially not any sort of viral stuff, or not enough of the viral stuff for it to actually kill that cell off. It just incorporates some new material. So it is a mechanism sometimes that we can utilize in biotechnology that allows us to manipulate organisms. I said sometimes, because usually we think about plasmids manipulating the plasmid, and with conjugation, it's easy to get that back into the cell because plasmids, like some plasmids, like that to occur, the conjugation. So it's easier to manipulate some of that, like that mechanism versus the transduction. We're not going into biotechnology and thinking about all of those details, but there are multiple ways we might be able to manipulate some stuff, all right? There's also other potentials on a little biotech note. We might be thinking about trying to manipulate other things. Instead of bacteria, we might use yeast cells. Yeast cells are eukaryotic, and maybe what we want is something more along those lines rather than bacteria. And we might manipulate things a little bit differently, all right? I'm bringing that up because as we move through here and start talking about some of the different features with virus, there are some special things that some virus have that lends to these potentials, all right? And we'll get to that in a second, all right? So, the flu virus, influenza, there are a handful where the virus, an RNA virus, will get into a cell and then move into the nucleus of the cell. Lots of RNA virus, and remember, a virus is either RNA or DNA. It's never both, all right? So those RNA virus, some of them, a few, do as we're seeing above, but most of the RNA virus, when it gets into a cell, it stays within the cytoplasm using the cells, the host cells, ribosomes, to read the RNA, and in turn to make the capsid and proliferate that way, all right? So in this case, we've got a handful of things. It's similar stuff where the virus makes contact with the specific type of cell. So a cell, within our mucous membranes is where influenza, where the flu virus would like to go. So it attaches to some of these, epithelial cells within the mucous membranes. As it makes contact, the spike proteins on the virus making contact with receptors on those cells, pulling it in through endocytosis, all right? And as we pull this virus in, you can see it's surrounded in a vesicle, all right? So it attaches, it penetrates in, being endocytosed, pulled in this vesicle. and then it uncoats. It basically breaks out of that vesicle, releasing the viral particles. This, in turn, lends to its opportunity to gain entry in, well, first off, it's gained entry right here, but to gain entry into the nucleus once it's been released from that vesicle. Now the RNA is able to get in there, start replicating, and in turn, making more the proteins that we need to make our capsid, plus more RNA to be packaged up inside, and then it's released. It's all, you know, attachment and penetration, the synthesis, assembly, and release, all of these things. The uncoding might or might not, dependent upon the types of cells. If we're talking bacterial phages, they don't do this. They don't need to uncode it, just inoculates it right in, all right? But when we think about eukaryotic stuff, they're going to be brought in a vesicle, and uncoding is a mechanism that needs to happen. Doesn't matter what type of virus we're talking about. They attach and penetrate, they synthesize, whether it would be in the nucleus or in the cytoplasm is kind of beside the point. But the flu virus is one that needs to utilize that. So that brings us to some thoughts about how this works, all right? And there are numerous potentials with the types of viral particles that can infect. As I said a second ago, as I'm sure I said before, it's one or the other, DNA or RNA. And when we look at the RNA, there are differences with it as well, all right? Here we're looking at the double S means single-stranded. So we're looking at a single-stranded piece of RNA that is the genome of that virus, all right? And there are some potentials with this. The RNA wants to be read by host cell machinery in the proper orientation, right? So the positive sense or the positive strand is the right orientation for the ribosomes to read this RNA. Similarly, like messenger RNA, right? We're going to read it in one direction. The negative strand is in the opposite orientation, right? So 3, 5, 5, 3, right, is what we're thinking about. The single-stranded is in the opposite direction, so we say it's negative sense or negative strand, and it needs to be converted into the positive sense before it can be copied, all right? So this is just showing there's some mechanisms, if it's single-stranded, for it to ultimately be manipulated in order to build the proper stuff. Shifting over this slide, and we're going to see some more details, a similar sort of image a little later on with some almost the same picture, but talking about some other potentials, how we may target some of this with therapy. So HIV wants to target other types of cells. We think about white blood cells, T cells, the T helper cells, all right, are really what's being targeted here. When we get into the immunology section after this exam, we'll talk a little more about some of these details, how it's getting in and the process that's occurring with that when we start talking about communication and T cells and so on. So that's what this is right here in the center, is a big T cell, and HIV makes contact. We can see the glycoproteins, the little purple spikes on HIV, making contact with a receptor on the cell. The CD4 is the receptor that HIV can recognize, and CD4 markers on these cells are indicative of T helper cells. CD4 and T helper are kind of it's the same thing, all right? It's just a class of proteins that allow for communication. It's truly not saying I'm going to try to snag me some HIV, but that's ultimately what happens in this circumstance. If these protein molecules are able to interact, it allows HIV to fuse to the cell surface, making contact and bringing it in. all right? So we can see this as it's bringing the virus in. As this occurs, the RNA virus, HIV, gets brought into the cell. We can see the capsid and the nuclear material being brought to the inside, all right? Similar idea like we showed with the flu virus, but instead of pulling it in a vesicle, this already has that envelope, right? It's got membrane on it and it fuses with the host membrane and it basically just allows it to dump in it instead of pulling it in through endocytosis. It's similar membrane, so it's easy to just fuse with that and then dump its payload to the inside, all right? So HIV gets in. As this happens, the RNA with the enzyme called reverse transcriptase, it reads the RNA and generates DNA, all right? So viral DNA through reverse transcriptase is formed. That's what we need to get in and hijack this cell, all right? There's a couple other things, and as we move through here, some of these things actually become targets when we think about trying to block the process of HIV being able to mature within the cell, if you will, all right? So if you can block multiple angles of this, you've got a greater opportunity of impacting the virus, all right? So once this has happened, reverse transcriptase makes that DNA copy from the RNA. It gets moved across the membrane. Another enzyme, there's multiple enzymes that play a role in this. An enzyme called integrase allows the DNA, the viral DNA, to gain entry through the nuclear membrane. And it in turn, with integrase, helps it to get into the host chromosome, all right? At this point, it is called a provirus. Once it's within there, it then now is behaving just like your own DNA. We unzip, we read it, and as we are making a copy of this, like we would be making messenger RNA, instead of making the messenger RNA, we make HIV. That's what we're making, all right? So we start pumping out copies of HIV. We also are making RNA molecules that are going to say, build me this capsid, build me my shell, right? All of these little kind of yellow dots around here are the capsid. We need to build that too, our little package to ship all of the nucleic acid out, right? That's this right here, these little bits, right? All of the stuff that we need to, and along with other stuff like the reverse transcriptase and integrase, all of that gets packaged up in our virus too, all right? That's this stuff, the little dots, all right? We package all of it up together. Here we can see it forming around it and then blubbing off. It's enveloped in the membrane of this cell. So we've got our proteins and then membrane material that surrounds it, that in turn allows it to merge and fuse with other cells like this. And so multiple things, and we will again start talking about ways that we may go in and try to target that. It is really pretty interesting to think about, and all sorts of advances in treatments and potential cures. I saw just the other day somebody maybe has actually been cured with some cord blood stem cells that ultimately maybe has cured that. I have heard of other things as well that like some possible bone marrow transplant from someone that has a specific genetic makeup that basically makes those people immune to all sorts of things. And if someone was not immune to that and had HIV and received this transplant, potential cure could be achieved. But that is, most people aren't going to just stand in line and say, but I want a bone marrow transplant, right? Probably not, right? So shifting over, chicken pox is something that, well, I'd like to say we've all probably had, but I bet you that's probably not true. A few of us have probably had chicken pox, and the rest of us, you were vaccinated, right? Not me, I had it, right? So that was one thing that I was like, should I get my kids vaccinated for chickenpox or just say, buck up, son, I had that, you're gonna have it too. They got vaccinated, right? My kids are like, I think you just want us to get shots. And I'm like, you're damn right I do, right? I want you to get any shot that you can get because I want you to be protected, right? It's not like I'm trying to inflict pain upon you. I don't want you to cry, but I'd like you to be vaccinated, right? I think it's good, right? So they just complain. Nobody wants shots, right? So I think if you could prevent this, you probably would want a shot, right? Are you saying shots like the bar? You're like, I want shots.
No.
It's early. It's barely 9, right? Holy cow. All right. It's fine. It's okay. I understand. You know, and if you're taking shots while you're studying for the test, you probably ought to take some shots while you're taking the test, right? It's state-specific studying, all right? Those are words to live by. Like, he told me to take shots, right? It just depends, right? If you have too many, you may forget that you're actually taking a test, right? That's bad news, right? Okay, so enough of that. And then you'll wake up and you're like, holy cow, I got chickenpox. No, that's not how it happens. Okay, so all of a sudden the pack back threads for this week, I'm gonna be like, what the heck did I say, right? How did this happen? Oh no, varicella zoster is the virus that causes chickenpox. It's a type. We will see all sorts of different types of viruses, and it technically is one that is super common, something that, you know, we typically think of as a common childhood illness, at least it used to be. And now with vaccinations, it's getting minimized for sure, all right? I remember back in the day being a little kid and there were chickenpox parties. It happens, typically you're hoping it's gonna happen right at the start of summer because you're gonna be out of commission for a little bit of time. One kid on the block gets chickenpox and it's like every mom knows. And all of a sudden there's a play date. We're going over to little Timmy's house to play, but Timmy doesn't want to play. He's laying on the couch sick, right? And you're like, why did we come over here? Now we know, right? We came over to get chickenpox because all you really have to do is walk in there and you're gonna get it, right? Done deal. Then you go home a few days later, you're laying there with chickenpox, right? You're like, what the heck happened? Damn you, mom, right? It all makes sense. You can't trust her, right? Not anymore, right? Not after you did that. All right. But it kind of makes sense, all right? You'd like to get it over with and not miss school, right? My kids are like, I'd rather miss school, right? It makes sense, right? So chickenpox, it's an envelope, so it's got host membrane around it, virus, and here we can see it with electron microscopy. It's double-stranded DNA, and it in turn incorporates into the host cell. This image is some shingles. It's not the same, although some of it sort of looks pock-like. They're more, you know, kind of little individual bits, and this is like big rashy patches. Sometimes like huge patches of skin that's all inflamed. And the virus, lots of virus, has the potential to kind of take up residence, go dormant, if you will, latent, on some nerve cells that lend to it coming back later on, all right? Varicella is a type of herpes virus, all right? And herpes, there are all sorts of different herpes viruses. I like to think of herpes as the gift that keeps giving, right? There are all sorts of potentials with that, and it comes back, it flares back up after some latency, and it depends on the specific nerve cells that they're taking up residence on, like with chickenpox, as far as where they may be flaring up. They have the potential to be all over, But when you get shingles, you don't think about it completely covering your whole body like you probably think about with chickenpox. Maybe you do. It's typically, systemic. It's all over where you see the pox, mainly trunk, but all over, right? And this, it's usually like a specific region, and it's this specific nerve that virus reactivated on, and that's why I'm seeing it right here on my side, or on this side, or my whole abdomen, right? Whatever. Hopefully not, right? So vaccines against chickenpox, vaccines against shingles for later in life, all right? And hopefully to prevent this from occurring, all right? There are, and usually when we think about it flaring back up, it's something that happens later, 60s plus, or when you start thinking about, oh, maybe shingles, and you could get vaccinated against that. It can happen earlier in life. It can be numerous things that may stimulate that, like a stressful situation could potentially do it, but other things like cancers may lend to some of that as well. So, a young adult with shingles is a red flag, and you want to kind of figure out what's happening here, because it's something that really shouldn't be targeting them yet, right? But we find out, oh, this is a college student getting ready to graduate. They're maybe thinking about graduate school or med school or whatever it could be, getting into the nursing program. There's all sorts of stressors, right, that potentially are enough to cause something like this to happen. So a little bit of investigating, and you're like, you're just stressed out, right? That's all fine and good. But I think if you knew, as you're getting ready to graduate, and now all of a sudden you're like, holy crap, do I have cancer? What's going on, right? Now that's the last thing, school is the last thing on your mind, right? But it's really probably just stress, all right? Oh, chat. Will Fridays be on the exam? Heck yes, it's gonna be on the exam. It depends on how quickly I get through this stuff, truly. If I can get through all of this, then we won't have lecture at all. But I bet you I'll still have a little bit of stuff, maybe. I can talk real fast. I know you guys probably want the day off. Want the whole week off. All right. So we're shifting back to bacteriophage for a second, but it is pretty similar when we think about other virus getting into cells. And it reminds me of a growth curve with just bacteria. If you think about something like that, there's going to be a little bit of time when stuff is just I just moved in. I'm just getting a feel for my surroundings, right? I just moved to Pocatello and I'm trying to figure out all of these one-way roads, right? Why, right? There's all sorts of things, right? That is kind of this, all right? So here we can see this is the log number of virons or virions, infectious particles, all right, virus. So as we look at this, we've got our inoculum, or a little bit of virus, whatever it is. This is a bacteriophage. So it's coming in contact with some cells, binding to these cells. This bit right here, this eclipse period, is where these, those crazy spaceship landers are on those bacteria. The bacteriophage is just making attachment and starting to inoculate or inject their nuclear material in. This is the penetration bit, all right? And so this eclipse period takes time for all of this virus to get in and do their stuff, all right? Once that has happened over the span of a handful of hours, now we've got virus that's inoculated or injected their nuclear material into those host cells. And this is this burst bit, the third one. So the host cells have been manipulated by this virus. It starts kicking the cell into overdrive so that all they're doing, all that bacterium is doing is making virus, all right? They copy the nucleic acid, they build these proteins, and it's starting to package it all up. And we're thinking like the lytic cycle with this, where we're going to destroy the cell. If we're thinking lysogenic, well, we'll get in, but we'll probably just hook up into the chromosome and lay in wait. And we're not going to think of all of the rest of this right now, but it's still going to happen once it does decide to be released. So the host cell releases lots of these particles. Bacteria are lysing, and dependent upon the type of bacteriophage, you're going to have varied levels of burst sizes. So you don't think of thousands of virus in a single bacterium being released. Handfuls, maybe 5, 10, maybe a dozen particles that are released from that cell, and there they go on, right? So there are ways we can look at this stuff. We can grow bacteria up on a petri dish and have it initially incubated with some potential virus. And if that virus is truly there and can target that bacterium, when they are initially placed together, this stuff is happening, right? They make contact and they're trying to get in. Once they get plated out and allowed to incubate, if that virus truly did infect those bacterial cells, as the bacteria is trying to grow on the plate, the virus is also killing some of those bacteria, and you're left with these little divots, these little holes that we call plaques. And the plaques are areas where virus has killed the bacteria. There's nothing growing there, all right? And a plaque assay allows us to determine burst sizes. How much stuff are we really looking at, right? And it's pretty impressive, all right, to see some of that stuff. And we will see a plaque assay a little bit later where we could see some of those holes within the lawn of growth, right? Within the bacteria. You in turn could go in and harvest some of that and kind of cultivate it, if you will, and get more of that growing. So in this case, we're looking at some researchers that are working with some Ebola. This is, I'm assuming, level 4 lab, BSL-4, biosafety level 4. We've got the space suits, we've got the hoods. In the hood, it's a negative pressure, so everything is going to stay within there. You're not getting air blowing out of the hood, it's sucking in, right? So it's negative pressure in there. I assume the whole lab is negative pressure as well, so the air is being dealt with in the whole lab. In case there's a breach here, the whole lab is that way, so none of that air is potentially going out, and their suits are the opposite. It's a positive pressure. They're all blown up, right? You don't want to hear it hissing.
How many levels are there?
4 levels. BSL-4 is the top.
Okay.
This is the one that I think, it terrifies me. If I was in that situation and I was leaving my house, kissing my wife goodbye, it really could be goodbye. Right? That's how I'm like, I don't want to do that, right? I think it's fascinating, but I don't want to play with Ebola virus, right? That is, I want to crawl into a spacesuit, right? Because you just don't know, right? And here you can see a few things. Gloved up, multiple layers of gloves. It's not just one glove, all right? Gloved up, and we also have taped them to our sleeve, so we are completely sealed up, all right? There's probably another glove and tape under that, and then the outer one. I'm not exactly sure, but they're definitely taped up on there. And like I said, it's all pressurized, right? All blown up in there. So crazy, all right, to think about that stuff. So we showed that tobacco mosaic virus at the very start, right? And I talked about filterable virus. It was one of the first, what was the first virus that was evaluated to be able to go through a filter. This isn't the same, but it's a similar idea. This is a type of filter that allows us to filter, sterilize things. Some stuff can't go through an autoclave, so we might filter, sterilize. And it depends on what we're looking for as far as the level of filtration, all right? So we've got a few things that are going on up here, all right? And here's an image of like a cross section of this filter. The little holes in there are the pores in the filter, and you can see a bunch of bacteria, and these suckers are huge. but they're not, right? But they look huge compared to the pore size. They can't get through there. So this filter will trap the bacteria and anything bigger than bacteria, but anything smaller can go through this little hole. Virus is considerably smaller, all right? So we've got a couple of different things going on over here. This filter, the pore size is 5 microns. Bacteria typically are smaller than that, a micron, maybe 2 microns. The green stuff is the virus, and the blue stuff is the bacteria, and the red stuff, well, maybe that's our blood, right? Maybe that's a red blood cell or something, a eukaryotic cell that's going to be bigger than 5 microns, right? So a 5 micron filter, and this is a syringe up above that's got the stuff in it. You hook the syringe up, you start squishing it or pushing it down, and the liquid comes through. Bacteria and virus can make it through that pore size, and we can see them both in that flask. We shift it up and use a different pore-sized filter that's 0.2 microns or 200 nanometers, very, very little holes compared to great big windows that the bacteria can get through. And now it's like this. The bacteria stay on the top side and the virus is able to go through, all right? Filterable, all right? Able to, too small, right? To be trapped behind. The first stuff when they were working with the tobacco mosaic, it's not like this. They have these filters that are like porcelain filters, glass, right? That seems like that should be solid, right? You shouldn't be able to get through there, but there is some porosity with that. It looks very odd, these filters, these porcelain filters. And you can have a membrane that you put on there. If we were going to test water, and there's all sorts of water quality testing. You hear all sorts of places where there are boil orders. Usually it's smaller towns. Rigby's had a boil order recently. I think Blackfoot a few years back. Yeah.
Our Christmas break, my hometown had a boil order.
Okay. Where at?
Haley.
Haley? Okay. So boil order there. And probably what they think coliforms in the water is what they say. That's E. coli or somebody else is really what they're looking at. Yeah, that's why I was like, I think Blackfoot, yeah. So sometimes it's issues with like, well, water systems and sewer systems and all of that gets put in decades ago and stuff starts to break down. There's all sorts of potential issues where things can happen like that. There are other problems, like if you live in some rural area where you've got well water, there are definitely potentials for that well water to be contaminated. It could be, you know, cattle down the road that ultimately contaminate the aquifer and lends to you getting ill. There's numerous things that set the stage for some of this, all right? So with water testing, it's like this, but they would have the big glass, the porcelain filter, they put the membrane on it, and then they'd have a big container on the top. It's hooked up, the bottom guy is hooked up to a vacuum pump, so there's a little glass nozzle coming off of that. And as you apply pressure to that, it's gonna suck the water through, all right? It would slowly drip through, but vacuum's gonna help it get through there, all right? Any bacteria will get trapped on that membrane. You then take that membrane, you unhook it all, you take the membrane and put it onto a Petri dish. That basically inoculates the Petri dish. And if it grows up, you guys are looking at the like selective and differential stuff in lab this week, right? You've got also, some of you are like, I don't know, but you will be, right? If you haven't been to lab yet. The EMB plate, eosine methylene blue, is the one that we typically utilize for water quality testing, and it will show up with different things on there potentially that will indicate that, all right? That's shifting away from the virus stuff, but it's a similar idea, right? As far as what we might be testing, all right? And all of these filters, basically virus is so small, we don't have, at least to my knowledge, a filter that's going to trap everything, right? Virus as well. If there is, I truly am not certain what that, I think 0.2 is as small as we get. And so we can see this over here. In these applications, we might be truly trying to select for this stuff, all right? And with my experience dealing with the C phages, last semester, we did similar things where we found some soil samples. We tried to get those to, if there were virus in there, we tried to really kind of ramp it up. So we incubated some soil with some broth that had some bacteria of interest in it. And if there really were virus that want to target that, well, now we gave them a target. We allow that to incubate for a couple of days, and the hopes would be that if that truly was the case, you're going to have a tube that's chock full of virus now. You take that liquid, centrifuge it, because you don't want to just put a bunch of mud in here. You centrifuge it, and now all of the liquid is up in the syringe. It's full of bacteria, and you hope it's full of virus. As you run it through the filter, the bacteria is removed. The virus, if it was there, stays in there, and then you test this liquid. Does it have it or does it not? And you use the same host bacteria that we started with to try to get it, the plaques, right? And if they're there, well, we're in business, right? And I see a few tuning in from home that are like, yep, I did that. All right, here's some plaques right here. So these, and who knows what, the details of all of these dilutions and stuff, but really that's what we're looking at, is a series of dilutions, a high concentration of virus, a little bit lower and a little bit lower. This is a bacteriophage T4, so it's like that spaceship looking one that I showed you, right? That's what T4 looks like. And it likes to target E. coli, right? So E. coli is all of this stuff. The opaque stuff is the lawn of growth. And the little divots, the little holes are where the virus has killed the E. coli and there is no bacteria, all right? So you can see over here, if my mouse will come back to me, over here it is just full of virus. There are holes all over the place, and then we can get down to lower amounts. Something over here, probably too many to count. This, you could count them, and you definitely could count that. And if you know how you diluted these things, you can back calculate and say, oh, this vial, this tube of liquid that's got virus must have, 1 billion virions or viral particles within it, right? And now we could maybe do some science with that, all right? One little virus, you can't do crap with that. But if you got 1 billion of them, that's a different story, right? We could extract their nucleic acid and do all sorts of other things, or take images and see them under the electron microscope. This over here is a cell culture. This would be for different virus, not bacteriophage, some animal virus, something that wants to be in an animal cell, not a bacterium, all right? And so they don't grow on a regular petri dish. They need living cells. And so cell culture has viable cells. It's got growth medium, the liquid that the cells are in, the nutrients needed for those cells to be maintained and stay alive, all right? Then, and these special flasks, you can, instead of a regular compound microscope, you could look at them under a stereoscope or a dissecting microscope. You put the flask right under the dissecting scope and you're seeing eukaryotic cells. And if they look weird, if their morphologies, or if they start getting destroyed one way or the other, it should be a nice monolayer of cells. And once we recognize it deviating from that, they're infected, they're dying or whatever, all right? And so you can evaluate that visually as well. Different, all kind of just different mechanisms with that. So we also may utilize other mechanisms to cultivate virus. The flu virus is definitely one of these ideas. There are others as well, all right? So eggs, chicken eggs, are a good sterile incubator for all sorts of things. And this is just showing where we may inoculate, dependent upon circumstances. And it's not going down to Albertsons and buying yourself a dozen eggs and thinking you're going to make yourself some flu vaccine, all right? They are embryonated eggs, all right? We've got a little baby chicken that's starting to grow in here. It's alive, all right? And we're going to use that to pump out some virus, all right? So we can in turn, as we are inoculating all of these eggs, we can create lots of virus. We go in and harvest that, purify it, and use specific fragments of that to build vaccine. Yeah.
The second picture, it looks like they're injecting into the different parts. How would they know, like, the proper part?
That's a good question. How do you know where you're injecting it? I would think they probably, so you can, there's all sorts of weird stuff with these eggs. You can cut little windows out. There was an embryologist that worked here years back that was studying development of all sorts of stuff. But chicken eggs were one of the main things that he was working with and manipulating them a little bit. And you could get embryonated eggs. He would get all sorts of eggs shipped to him. All of a sudden, you go to the store, you see like the 5 dozen eggs, big thing. And you're like, holy cow, that's a lot of eggs. Whenever I see people getting 5 dozen eggs, right? I'm just like, that's a lot of eggs, right? Awesome, right? Too much, right? He would get stuff like that, but they're not Albertsons eggs or wherever you're getting your eggs, embryonated, right? And then he would take them up there and you could make with a little scalpel, you can cut windows in there. Then you put a piece of tape over that window and you can watch the development. There's all sorts of things. So I'm assuming they probably know, as they have made these images before, where we need to go, right? I don't know. I don't know if they've got a camera that they're going in there and looking at, probably something like that where they could see it. This doesn't look like that. They're just inoculating it in, right? And maybe in this circumstance, doesn't matter, right? As long as you're getting it in there. I don't know for sure. I would think it probably matters with some of it where you go to the yolk or to the actual embryo or whatever, right? But again, I don't know. So as this happens, wherever you've inoculated it, and however you figured out that that's exactly where you want to go, you grow this up within that egg so it produces good amounts of virus, and then we go in and harvest that out, right? This little chicken's going to take one for the team, right? And we in turn can harvest and purify and produce vaccine that way. So the flu vaccine is generated this way. There are definitely some with biotechnology, some ideas that maybe we could switch this up. With the H1N1, the swine flu or Spanish flu stuff that we had as, issues, when was it, early 2000s, when that was happening. That wasn't in the normal flu vaccine, but they quickly produced some vaccine, all right? And they quickly produced it in a different mechanism. They didn't use chicken eggs because that stuff, in order to do it, takes months, all right? It takes a super long time to get it to grow up and purify and whatever. They took a specific chunk of material, the information to make this specific spike protein, and with some bioengineering, they incorporated that into a plant, tobacco plant. They grew the plant up. As that plant is growing, it's producing this protein. They harvest it, chewed it all up, and blended it up, and then extracted it out. And then they got all of these proteins. That protein was what was utilized to build that vaccine, all right? There's all sorts of different mechanisms and different ways. We're seeing more stuff like with COVID and the mRNA vaccine, different, right? That, not the mRNA, but using a plant instead of a chicken sets the stage for alleviating issues of, people with, chicken allergy or egg allergy. They're not going to have issues with that, and they could get the flu vaccine. But otherwise, you're not going to take it, right? Just kind of depends. This goes back to kind of this cell culture, right? As we're looking at this liquid, all right? Here we can see these primary cell lines and then continuous cell lines. It depends on the circumstance, all right? So this, we've got lung tissue, individual cell isolated, a primary cell culture. These cells start to grow. You've got a few of them in here, they start growing. And once they've come in contact with all of their neighbors and the edge of the dish, whatever it is, they stop growing. Contact inhibition is when a normal cell makes contact with another normal cell and they're like, hey, how you doing? And they just stop, right? That's what you want. And that's what we want in our body, all right? This requires, with normal cells, requires repeated inoculation, where you take some of this and put it into a new culture, because once they get to this point, they're not growing anymore. I mean, they're alive, but they're not growing, right? So we just keep culturing. The other one is different. And this continuous cell line, this lung tissue is cancerous tissue. And this continuous cell line, contact inhibition is no longer present. And we've got a petri dish overflowing with these cells, all right? And so it's something that is immortal, if you will. It doesn't, cancerous cells don't know how to die, and they continue to proliferate. The HeLa cells are from Henrietta Lacks. She donated or was robbed of these cells one way or another. They got these cells and it is used, it's been used for decades for all sorts of research, more than just cancer research, as a continuous cell line. Kind of crazy stuff. So this is it, the HeLa cells, all right? So you can see these cells and some of the protein within that. And again, there's all sorts of details with that and information about her that I think is pretty fascinating. All right, guys, I will leave you with that. We'll pick up the littlest bit. How much do I have left in here? I need to stop sharing first. Oh, 10. Did you say 10? You're on top of stuff. Okay, so we'll wrap up the littlest bit of this Friday. I can answer any questions if you've got any about the test. I have not finished building it yet, but I've got it started. Okay, guys, have a great day.